let's talk about how we can control the cell cycle. The cell spends most of its time within interphase, which is split into three parts: G1, where the cell does most of its growth, and then S phase, where the DNA replicates, G2, where it prepares for mitosis, so condensing up the chromosomes, for example. And then in M phase, which is the mitosis phase, and followed by cytokinesis, the cell divides into two new daughter cells, which then continue following the cycle. Now there's also this G0 phase, which is, or G0, I don't know, but that's when it comes out of the cycle, and that can be, say, for neurons, when they're terminally differentiated, they don't actually divide anymore, so they kind of step out of the cell cycle for a bit. Some cells can actually go back into the cell cycle after being out or paused for a bit, so that would be like liver cells. So we're going to talk about how we can control the cell cycle. So first of all, a very important molecule that helps us control the cell cycle at different checkpoints is cyclin-dependent kinase, which pairs up with cyclin. The first duo of this that was found was mitosis promoting factor, and it is a heterodimeric protein. Dimeric made up of two things, hetero two different things, which are cyclin-dependent kinase. In this case, it was cyclin-dependent kinase. There's different types. We'll talk about those soon. And then there's the cyclin. So the amount of Cdk, or cyclin-dependent kinase, found within a cell is always constant, but then the amount of cyclin is what fluctuates, and that's how you control the activity that you want going on in the cell cycle. So if we look at the concentration of cyclin throughout interphase, it's fairly low, which means even you do have Cdk, but it's not going to work because there's no cyclin attached to it, so it's not going to be able to do its job. Since it's a kinase, it phosphorylates things and keeps the cycle going. So it's not going to undergo mitosis if there is no cyclin present. But then as cyclin concentrations increase, Cdk activity increases, and then you can do mitosis. And then again, interphase, cyclin concentrations decrease, Cdk is inactive, and so on, as long as you are completing the cell cycle. There are many different types of Cdks, and they each work at a different step within the cell cycle. So first of all, there is the G1 Cdk, which helps the cell get through the G1S checkpoint or restriction point. We'll talk more about that one in a later video. And then after that, there's the G1S Cdk, which pretty much tells the cell, okay, now you're going to go replicate your DNA. And then there is the S-CDK, which works in the S phase to actually help replicate the DNA. And then there's the M-CDK, which, as its name implies, the mitosis promoting factor, promotes mitosis and mitotic events.